Good morning. My name is Anita Chetty. I'm the Upper School Science Department Chair. I'm also the advisor for Harker's Women in STEM program. I'd like to welcome you to our 17th annual Harker Research Symposium. The number 17 is important because when you think about the life of a child, when they're 17, they're probably close to the end of their K-12 journey. And what does that mean? Well, if they are right now a junior or a senior, i.e. grade 11 or grade 12, that means that they might have come to the research symposium when they were just a youngster. Maybe they came to play with magic sand. Maybe they came when they were in middle school to share their discoveries with their friends. Or maybe when they got a little bit older, they came to inform and to empower their youngster friends who are now in middle school or in lower school. So you can see that for 17 years, the Harker Research Program has been instructing, inspiring our students. And what I really love to think about is that we all come to be inspired by the entrepreneurs, the scientists, and people like Janice who are doing things that are going to improve our lives in the future. Just a couple of program notes before we get started. We do have a first aid kit that is at registration, which is where you came in. And a reminder that the lunch is in the quad. We're very lucky, the weather is cooperating. There's also a science magic show in the quad. We do have signage to let you know where everything is. I do want to take this opportunity to thank Wisdom, our chief organizer of this function. You can only imagine the number of hours that have been invested to put this event together for you. The Harker Research Symposium is a legacy, and I think you being here, I'm so appreciative because we are continuing on the tradition, and Wisdom is a big part of that. So I want to thank the members of the Parent Outreach Committee that worked so hard since October to get exhibitors for us. For the first time, we have 12 exhibitors in the Nichols Atrium. Please make sure you go and check them out. And also, I want to ask you to think about the people who work behind the scenes as you walk around the campus today. The grounds look so lovely. The food that you're going to eat, the tables that were set up, all of that was done by people who work all the time. So today is a day of excellence, not only for our students, but it's also a celebration of Harker excellence and all the people that work here behind the scenes. So thank you so much for joining us, and without further ado, I'm going to turn it over to my Wisdom co-president, Eileen Ma, who will introduce this morning's keynote speaker. Thank you again for joining us.
Hi everyone, thank you Ms. Chetty for that wonderful introduction. So as you guys know, I'm Eileen Ma, one of the co-presidents of Harker Wisdom this year. And before we get started with today's events, I just wanted to take a moment and give a huge thank you to Ms. Chetty. So let's give her a round of applause. In the past 17 years, this symposium would not run without her tireless efforts and enormous dedication. So thank you so much, Ms. Chetty, we appreciate you. All right, before I introduce today's first speaker, I have a couple short requests. Please kindly silence all of your cell phones so that way it's not distracting to our speaker and our fellow audience members. Additionally, there are no food or drinks allowed in the RPAC. I promise you guys we have enough snacks, so please leave all of them outside for after the talk. And finally, please place all of your water bottles away. So with that out of the way, it's my absolute honor to introduce our opening keynote speaker of the day. Dr. Janice Chen is the co-founder of Mammoth Biosciences, a biotech company that's harnessing the power and versatility of CRISPR technology to eradicate genetic disease. This is a company she founded with Lucas Harrington, Trevor Martin, and her former PhD advisor, Jennifer Doudna. And yes, she is the Nobel laureate who developed CRISPR. After earning her bachelor's degree at Johns Hopkins, Janice went on to receive her PhD in molecular and cell biology at UC Berkeley. From there, she has gone on to achieve some incredible honors, like making the 2019 Forbes 30 Under 30 healthcare list. Dr. Chen is truly an incredible scientist and human, open to use the power of CRISPR to better humanity. Today, she will be giving her speech, Reading and Writing the Code of Life, detailing her own journey through STEM, her path to entrepreneurship, and her vision for what the future of gene editing entails. Please put your hands together and give a warm welcome to Dr. Janice Chen.
Good morning. Thank you so much to Anita and all the organizers of today's symposium for giving me the opportunity to speak with you today. Just to let you know, I'm recovering from a bit of a cold. My daughter just started daycare, so I'm learning the realities of getting hit by all kinds of viruses. So I don't believe I'm infectious, but if I cough, I just wanted to give you that heads up, so I apologize in advance. So today, I will be talking about a little bit of my own personal journey. We'll be sharing, some of the things that I did leading up to starting graduate school, starting Mammoth, and the work that we're doing at the company to really enable the full potential of CRISPR technology. I wanted to start by first talking about why it's such an exciting time to be in life sciences. As you all know, advances in sequencing technology has really paved the way in helping us understand the relationship between our genes and our health. For instance, sequencing of the human genome has really opened up new avenues for really understanding, the basis of genetic disease and what we can do to treat those areas. We've also been able to train our immune cells, for instance, in immunotherapies to specifically fight cancer cells while avoiding the healthy ones. And as you all know, CRISPR is one of those breakthrough technologies that's really making a difference in research and development from early research all the way through clinical medicine. And so I'll also share a little bit about what CRISPR is and how we're using to deploy it to detect and cure disease. So a little bit of background on CRISPR. What does CRISPR stand for? Clustered, regularly, interspaced, short palindromic repeats. It's a mouthful. You don't have to remember it, just that's what it stands for. I can go a little bit into the biology, but fundamentally, CRISPR-Cas systems exist in nature, and these are adaptive immune systems that remove foreign DNA and RNA. In the same way that our bodies fight viruses, bacteria have the same types of immune systems to fight against these foreign foreign viruses that invade these bacteria. And it's a very incredible system that is able to use these Cas proteins that actually target those invading viruses and cut them so that they no longer produce an infection. So just again, a very high-level introduction to CRISPR biology. Bacteria have evolved diverse defense mechanisms to protect against foreign DNA and RNA. There's a lot of different ways that bacteria do this, and again, a lot of these have been through evolution, very, very effective ways against fighting foreign DNA and RNA. One example of doing that is through physical barrier. In the same way that we have, skin as a physical barrier to fight against infection, bacteria have, their own mechanisms of doing that. There's also restriction modification systems. This is another method for, cleaving foreign DNA sequences. There's Argonaute proteins. This is another mechanism for cleaving RNA. And of course, CRISPR-Cas, which is one of the most exciting discoveries that have been made in the last decade, and how we've been able to repurpose this natural immune system for genetic editing. So the biology of CRISPR-Cas, again, enables bacterial adaptive immunity. This is an overview of how CRISPR and its use in bacteria have been repurposed for gene editing. So as I mentioned, there's 3 phases of CRISPR-Cas bacterial immunity that exists in nature. The first is adaptation, the second is expression, and the third is interference. And I'll walk you through just a little bit about the different steps in how this is done in the native bacteria. When we talk about adaptation, this is the first step. This really means the acquisition of foreign nucleic acid sequences in CRISPR arrays. What this means is when a virus initially infects that bacteria, there are mechanisms within the immune system that actually are able to extract those foreign nucleic acids and integrate them into these CRISPR arrays. Again, remember, this stands for the clustered, regularly interspaced short palindromic repeats. And very excitingly, this was initially how the CRISPR systems were discovered, because scientists saw these repeats that are indicated by these black triangles that existed in these bacterial genomes. And they asked, what is these repeats? What is their function? And is there something unique about them? So that was the first hint that there was something that was quite interesting. And this is where scientists were able to probe a little bit further to really uncover this really elegant and process. So once those foreign nucleic acid sequences are integrated into those CRISPR arrays, the next step is expression. And this CRISPR array is used to generate what's called mature guide RNAs. And we'll talk a little bit more about that. So what happens here is that CRISPR array gets transcribed from the DNA to the RNA. These are called precursor guide RNAs. Those precursor guide RNAs get processed so that they become mature RNAs. As you can see, they become shorter segments of RNAs that are then able to guide the CRISPR-Cas proteins to that target sequence. So now if you think about the sequence from the foreign virus that was put into this CRISPR array is now transcribed into an RNA that is used to guide any future CRISPR-Cas nuclease to that target sequence. So anytime that bacteria faces a new infection from a virus that it was previously exposed to, the bacteria is able to use its CRISPR-Cas immune system to eliminate that infection. So this is a really powerful way that bacteria have evolved to manage new infections. And as mentioned, the third step, interference. And this is when that Cas effector protein is bound to that mature guide RNA. That mature guide RNA is complementary to that target sequence. That cutting event happens, and now you've eliminated the virus. So this is the biology of CRISPR, and one of the most exciting discoveries, as we talked about, is the fact that this CRISPR process can be reprogrammed for gene editing. And this was the first application demonstrated with CRISPR, and the work that had received the Nobel Prize in 2020 by my former PhD advisor, Jennifer Doudna, and colleague Emmanuel Charpentier, for their work on CRISPR gene editing. So as I mentioned, the guide RNA is able to bind with the Cas protein. That particular guide RNA can be designed to target any sequence of interest. This is both DNA and RNA. Upon recognition of that target sequence, this triggers cutting of that DNA or RNA sequence. And then if this happens in a cellular context, repair mechanisms are then able to trigger a gene editing outcome. And there have been many ways that scientists and researchers have been able to adapt this fundamental process to do really, really interesting things with respect to modifying the genome. And I'll talk a little bit about that as well. So today, CRISPR is eating the world. When I first started in the lab, CRISPR was quite a niche area. A few people were familiar with the Cas9 protein. I think even fewer people were familiar with even the fundamental biology of CRISPR. But over the last couple of years, CRISPR has really opened up our imagination because of its potential of really treating the underlying cause of disease. And of course, with DNA encoding the instructions for life, being able to have the ability to modify the genome has also opened up new opportunities for thinking about how we can leverage a gene editing tool to better humanity. So these are just examples of articles that have been written. These are some early journals, actually. So you can see, CRISPR is changing life as we know it, editing humanity, CRISPR everywhere. We can now engineer the human race, the DNA revolution. So again, this is a very, very exciting time as we think about how technology can interface with biology and what that means, not just for our species, but also, planetary health. Again, the possibilities are tremendous, and this is a big part of what I think the future will look like as we have even more precise tools for gene editing. So I'll talk a little bit about my journey to graduate school. I was actually born in Southern California, but I grew up in Salt Lake City, and this is a photo of my family. I am one of 5 kids. My parents immigrated from China before I was born, so it's been quite an exciting journey, I think, for my family to grow up here in the United States, but also have really strong values from my parents as they grew up in China. My siblings and I have always been raised to really do our best in everything and really take control of all opportunities that come our way. That's something, and those are values that I've really taken to heart in everything that I do in my own life and what I hope for everyone. I think I'm really, really grateful for my parents who have opened up so many doors for me and really, pushed me to reach my fullest potential, and that's my greatest hope for everyone here in this room as well. Some of you may know my youngest brother is Chen, 2022 Winter Olympic gold medalist in men's figure skating. I think that was just an incredibly just emotional time for all of us to see how just years and years of hard work, dedication, sacrifice, and this full support of a team, and the ingredients of all of that, and what that can really allow my brother to achieve. So I think that's been something that, of course, my family carries with us all the time as we think about what it takes to be a champion. And that can be, a champion can have many different types of flavors and outcomes, but to see the ingredients and what it takes to do that is something that's really motivated me, and I hope I can, inspire others as well. So, my dad also was a PhD student at the University of Utah, and that's one of the reasons why we relocated to Salt Lake City, and he was studying pharmaceutical chemistry. And so, and when I was very young, he was in the laboratory. So even as a really young child, I was exposed to molecular biology, being in the lab, before I really even understood molecular biology. I was playing around with test tubes and pipettes, looking at gels, and it's quite funny because growing up, instead of having photos of the family on the fridge, there were photos of gel images that showed some of the experiments my dad was running in the lab. So even at a young age, I learned about recombinant DNA, probably earlier than I would have expected. But I think that exposure really helped me get a deep appreciation of how fundamental science can lead through exciting technologies. And I think I had a very kind of natural affinity with STEM. And that was something that I credit my dad in terms of giving me that early exposure, but also really fantastic teachers in elementary school and high school who really cultivated that interest and allowed me to push myself to pursue those passions. And that was a big driving reason as well for me going to Johns Hopkins for undergraduate. I felt that Hopkins had some of the world-class biomedical research and incredible opportunities for helping young scientists like myself have scientific freedom to join labs, to pursue research projects. And in fact, early in my undergrad career, I started a course called Build the Genome that was run by Dr. Jeff Buka with the vision of being able to rebuild the yeast genome from scratch. So the idea that you could start from sequences of DNA and bring them together to produce a living organism was something that was truly mind-boggling for me. And I was able to be part of that course and learn about, again, all the basics of molecular biology, of cloning, and being able to pull together these building blocks to make chunks that became chromosomes, that became part of the yeast genome, so that it could be a viable organism at the end of this project. And once we understood how to build a viable organism, we could think about how we could build a minimal yeast genome to really understand what are the core components to enable living yeast organisms to thrive. And so that was a very exciting time for me. I was able to really get deep into the science, and that also then opened up doors for me to start in a undergraduate research laboratory with Dr. Bob Kulada, again now studying the genetics of yeast and the impact of superoxide radicals in yeast metabolism. So all of these different opportunities, again, just following my passion and interest in science and in the scientific method, and following mentors who really were able to coach me and push me to think to be an independent scientist. After my time at Hopkins, I was really fascinated by this idea of synthetic biology. I think, again, based on my early exposure to building the yeast genome, to then understanding the genetics of using yeast as a model organism, I realized that there was incredible potential in being able to modify all kinds of organisms to do very interesting things. So for instance, at Harvard, I joined the lab of Professor Pam Silver, who's a leader in the synthetic biology space, and she's always had really blue skies ideas around how can you encode for memory in bacteria, how can you modify bacteria that only depend on certain environments to produce biofuels. And so I was able to get experience and exposure to a lot of really exciting projects. But also I learned that there were a lot of challenges in the genetic tools that were available at the time. And so I was working with a particular bacterium which used pretty old school technologies for modifying a genome. And at the time I only had a very surface understanding of CRISPR. I knew that this was a gene editing tool, but at the time we weren't using it. We were using some pretty crude tools to be able to manipulate this bacterial genome to produce biofuels. So that was a very interesting learning that made me realize in order for us to build even more impactful technologies, it's important for us to go to the fundamental mechanisms of how these tools actually work. And that was a big motivation for me ultimately to go to graduate school at Berkeley. Graduate school, for me, was a really, really incredible time. And this was back in 2012, in 2014, when I started grad school. And at the time, the work that I just talked about with CRISPR and its use in gene editing was just first demonstrated in 2012. So this was a time when there was a lot of unknowns about CRISPR, but a lot of excitement about what it could do. And I was completely enthralled by the opportunity in Jennifer Doudna's lab. And at the time, Jennifer's lab was really focused on the fundamental mechanisms of how CRISPR proteins work. Again, there were lots of unknown questions, and I realized being able to understand the fundamental science was the way for me to really be able to enable new technologies by learning how these enzymes work. Science takes a village. And in my time in the Doudna lab, I realized that there were just a ton of really, really smart scientists, both in the Doudna lab and all over the world. And one of the great things that Jennifer brought to her lab was a spirit of collaboration and working with other people to solve very difficult problems. That was one thing that everyone in the Doudna lab embraced, and something that made me just thrive in graduate school. And I think there was a bit of an unfair advantage because if I wanted to collaborate with anyone in the world, I would say, hi, I'm from Jennifer's lab, let's collaborate on this problem. And almost 100% of the time I would get a response, yes, let's do it. So definitely, took advantage of that opportunity, and this is just examples of many different labs that we worked with. First, we were just trying to understand the fundamental mechanisms of how Cas9 is able to cut an on-target while avoiding off-target editing. And then based on those understandings, can we engineer the Cas9 protein to be more specific for editing applications. We also then worked very closely in looking at enzymes beyond Cas9. There was a whole explosion of new Cas enzymes that were being discovered, and through the again mechanistic process, learned that there were some CRISPR enzymes that not only edited DNA but were also capable of detecting these DNA and RNA sequences, and so that opened up new applications for diagnostics. And so I've had a very exciting time in working with Jennifer. She's been an incredible mentor and has really pushed me to think about what is it that I really want to do. I remember an early conference, this was near the end of my graduate school, and I was thinking about, well, what's next? I was thinking about, maybe going to industry, maybe, thinking about the impact that I can make in other companies, and Jennifer just sat me down and said, what do you want to do, and are you pushing yourself hard enough? And I think that was a really eye-opening moment, and to have mentors really challenge you and say, look, you have incredible potential, just go for it, don't be afraid of what you don't know, we're here to support you. I think that's something I feel incredibly grateful, and I wouldn't be here today if Jennifer hadn't pushed me in taking those risks, both scientifically and in my professional career. So many of you who've heard of CRISPR have probably heard of Cas9, and again, Cas9 is just the beginning. This is what Jennifer and Emmanuel received the Nobel Prize for in 2020, again, for the demonstration of gene editing using this Cas9 protein. But one of the very exciting things we learned during our time over the last several years is that, again, the diversity of CRISPR goes well beyond Cas9, And we were quite lucky as a field that the first protein that we discovered and worked to understand actually had very robust editing properties. However, over the course of 2015, 2016, 2017, et cetera, there was new families of Cas proteins that were discovered. Examples here like Cas12, Cas13, Cas14. These all look very different from Cas9, had different mechanisms, and through that understanding, we learned that these new CRISPR proteins actually have unique properties that are not found in the Cas9 system. And some of the examples are listed here. Some of these enzymes are much smaller, they have different types of edits, they're able to do nucleic acid detection, they have different requirements for targeting, they're able to do multiplexing, there's higher fidelity, and they have different levels of immunogenicity. And so all of these attributes are really important when you think about what is the use case for CRISPR in different applications. So again, my conversation with Jennifer was, I would say, a turning point for me in thinking about, let's build something new. We had developed and were inventors of a lot of these technologies at Berkeley that had tremendous potential and I really had this drive to push these technology outside of the academic lab. And we felt one of the best ways of doing that was to build your own company, write your own rules and just take the risk of making this happen. So as I mentioned, I started my PhD in 2014 in the Downhill Lab at Berkeley, and then graduated in 2018. And during the last couple months of graduate school is when I partnered up with my lab mate, Lucas Harrington, and as well as a colleague from Stanford, Trevor Martin, and with Jennifer together, we decided to co-found Dynamic Biosciences with the idea of expanding the potential of CRISPR and harnessing the diversity of this technology platform to detect and treat genetic disease. So leveraging all of the inventions that we had made over the last several years, really proving the thesis that you could go from CRISPR discovery to characterization to application was something that I realized we had been proving out in graduate school, and the next step was being able to start a company to even push this concept further. So, as I mentioned, Dynamic was founded to deliver on the promise of CRISPR. And we don't think of CRISPR just as Cas9 and just as gene editing. We take a broader step, leveraging CRISPR's fullest potential, and we think of this as a search engine for biology. As I mentioned, that guide RNA is sort of like a search bar where you can type in any genetic sequence of interest. So whether it's, let's say, Huntington's disease, SARS-CoV-2, cystic fibrosis, EGFR, these are just examples of different sequences that you can modify the guide RNA to specifically target using CRISPR. And once you're able to target those areas of interest, these Cas proteins will then perform a specific function to provide a gene edit outcome. And so those outcomes could involve reading that sequence. So in the case of detection and diagnostics, this could involve writing those areas on the genome to correct a faulty gene. You can knock out genes. You can modulate the gene expression. So a lot of really creative ways that CRISPR allows us to modify that DNA or RNA sequence that goes well beyond just cutting applications, which was the first demonstration of what CRISPR could do. So over the years, it's been tremendous growth at Mammoth. We started the company in 2018. We raised our Series A and had about 11 people in the company. We were based in Dogpatch in San Francisco in an incubator space where we shared a lab with many other startup companies. And so it was quite a fun environment to be among lots of other small startups trying to figure out their business models, trying to figure out, how to make the science work, how to bring in investors to further the mission of not just our company, but of course all the companies that were in that space. At the time of our Series A, again, we had brought in that diverse CRISPR toolbox based on the inventions that we had done in the Downhill Lab. And so we were exclusively licensed to IP. This includes the compositions of these different enzymes, as well as different applications like detector for diagnostics. And over the years, with our Series B, growing the team to 35, we're able to prove out some of these technologies and validate them with partners. Same thing as we brought our Series C in 2020. As we all have lived through some of the challenges of the pandemic, it was no different for us at Mammoth, where we had at the time recognized that we could pivot our diagnostics platform to target SARS-CoV-2, and this was a time when there was no name for the coronavirus, and it was quite scary because we had to make some serious decisions about, enabling the safety of our workforce, but also making sure that we had the opportunity to continue to push forward the development. So at this time, of course, Jennifer won a Nobel Prize. We had received some major government contracts and published a landmark paper demonstrating the use of CRISPR for detecting SARS-CoV-2 in patient samples. Many of us work with wonderful collaborators in pushing that forward. And now, most recently, we raised our Series D. We've grown to a team of over 150 employees and continuing to push forward our broader vision of CRISPR and have landed some major therapeutics partnerships with folks like Vertex and Bayer to really accelerate the development of our ultra-small CRISPR nucleases to the clinic and continue to push forward our internal pipeline for diagnostics and therapeutics and expand our IP portfolio to really enable the potential of CRISPR. So zooming out, how do we think about discovering and engineering these past enzymes? It really starts with what nature has already done. As I mentioned, these CRISPR systems have evolved over billions of years in nature, and nature is a really strong driving force in being able to generate nucleases that have incredible properties that sometimes humans cannot even think of. And so we go to these natural environments and use a process called metagenomics, which allows us to go into environments like hot springs or lakes, rivers, extract the natural diversity in terms of microorganisms that live in these environments. And as I mentioned, these bacteria all harbor various kinds of CRISPR systems. So we can extract that genetic material from these diverse environments. We're able to then take those sequences and put them in a computer. And then using bioinformatics and machine learning, we're able to take from those sequences, then identify specific CRISPR-Cas proteins. Then we reconstitute those proteins in the lab, and then we're able to screen them to understand how they work. And then through that whole process, when we understand those characteristics, we can use them for a number of different applications, such as the ones listed over on the right, biomanufacturing, diagnostics, therapeutics, agriculture, research tools, and much more. And so you can see that there's just really a huge opportunity for, not just the toolbox of nucleases that can drive forward these applications, but also all the things around that nuclease that must happen in order for us to build products. So that for the therapeutics application that involves delivery technologies, how do we actually get those CRISPR enzymes to specific tissues and organs of interest, and also other types of modalities. So beyond just cutting the genome, can we write new genes? Can we change the expression? Can we change single bases? These are all key areas that we work on within Mammoth to really open up the potential of this technology platform. So we think about what's the roadmap for therapeutics, and one of the really key breakthroughs that we drive for is how do we unlock in vivo genetic cures. Right now, the field of CRISPR, although has tremendous potential, is still limited in terms of getting these genetic medicines to the right areas. A lot of the current clinical trials and research and development focus on indications where you're able to extract cells from the body, provide that CRISPR edit, and then return those edited cells back to the patient. This is called ex vivo editing, and so this is through different examples that you may have heard of, like iPSCs or CAR T for immunotherapy, and these are very powerful medicines. However, they still are, very, they require a lot of effort in terms of, the patient journey, and being able to do the right delivery is quite expensive. It's very burdensome for the patient to go through this process. And the future is being able to deliver these nucleases directly to the patient and have those target the specific organs that are needed in order to prevent or correct genetic disease. And that's the future of in vivo therapeutics. As I mentioned, a lot of the work that we are doing as well is partnered with some major biopharma that bring in complementary expertise that help us continue to drive the development. So again, with Vertex and Bayer, but also we are working internally on specific indications as well as novel editing modalities to continue to drive forward this opportunity. As I mentioned, one of the key things that we discovered through our toolbox of CRISPR enzymes is the fact that some of these enzymes are incredibly small. And at the time, the field sort of dismiss these really small proteins. The thinking was, if these enzymes are small, they're not going to work. And that was one thing that we disproved during our time at the Down lab, and together with my co-founder Lucas, we're able to show that just because they're small, actually doesn't mean they don't work. And one of the really exciting things we found was not only do they work, they also work as good as the Cas9 system. And because they're small, it opens up a lot of advantages we'll talk about in a second. The first is, as I mentioned, delivery. Delivery is a major blocker, and right now there's sort of two major categories of how genetic medicines are delivered to the body. First is LNP, so many of us are familiar with lipid nanoparticles, and this is able to package mRNA. As many of us have received the COVID-19 vaccine, the same idea around delivering those molecules to patients. And the second general category is viral delivery. So this is through AAVs. And both of these delivery modalities have their attributes and the challenges. For AAV, the main advantage here is that you can actually have targeted delivery to areas like muscle, CNS, the lung, etc. But the big drawback is that the viral vector itself, has a limitation in terms of what it can carry. And in the first version of the Cas9 protein that was demonstrated through gene editing, what we're showing here is that it's quite a large molecule. So, 1,400 amino acids, which, as you can see, is sort of hitting the limit of what an AAV can actually package. However, with these ultra small enzymes, not only can you fit this Cas protein within these vectors, but you can also bring in other payloads for precision editing applications. So again, not just cutting the genome, but actually doing precise genome surgery in a very targeted manner in target tissues and organs. So this is very enabling for thinking about what the future of genetic medicine will look like. And again, these capabilities go beyond science. We have enzymes that have expanded targeting range. There is a particular sequence within CRISPR for CRISPR-Cas targeting, which sort of defines that zip code for where a Cas protein can land. It's called a PAM sequence. And with some of the original enzymes, as you can see in the blue bar to the left, if you look at the full targetable sites in the human genome, it's actually quite limiting because this PAM sequence is restricting all the sequences that can be targeted by the CRISPR protein. However, in looking at the diversity of CRISPR, we've been able to identify enzymes that have very, very sort of promiscuous PAM sequence, which what that means is that we can open up new sites within the human genome to target sequences that previously were not able to be interrogated with the older enzymes. And so, again, you can see a lot of the human genome now being accessible through these new enzymes. And finally, as I mentioned, these small enzymes and these new enzymes are robust editors. Cas9 has really exceeded our expectations in its capabilities and its activity, but Cas9 is just the beginning. And with these other enzymes, small enzymes like Cas9, we've been able to demonstrate in T cells that not only can you get comparable editing, in some cases it's slightly better than Cas9, and is showing the activity in T cells. We've also been able to show robust multiplexing, meaning that we can knock out not just one gene, but also 2 or 3 genes, and are able to maintain pretty high levels of editing with that approach. And so this really opens up exciting areas for things like doing a one-step editing for creating universal CAR T cells for immunotherapy. So as I mentioned, one of the very exciting opportunities is being able to deploy CRISPR to permanently cure genetic disease. This is an image that shows some of very well-known genetic diseases that are very serious and devastating for patients who live with them. And one of the advantages of CRISPR is that we can go in and actually correct those genes in a way that is curative for these patients. In addition to the delivery, the other challenge for genetic medicine is understanding the biology of different genetic diseases. But what's exciting is that there's still thousands of genetic diseases that we do understand exactly, the area within the genome that needs to be corrected for us to provide a therapeutic effect. And so this is just, again, a list of those diseases that could be potentially cured using CRISPR. So one of the things I'm going to end this talk on is, again, the power of the CRISPR platform. We started Mammoth because we knew that CRISPR was going to have huge implications beyond gene editing, and one of the areas that we were pioneers in is this diagnostics area. And again, the same process of discovery to characterization has opened up the recognition of enzymes that can detect DNA or RNA sequences and provide a new method for molecular detection. And in the same way, as I described previously, these enzymes are just the core machine for diagnostics. There's all the things that wrap around this in order for these CRISPR proteins to be used in the lab, point of care, and point of need environments in order to think about how the current molecular diagnostic tools like PCR can be leveraged in other settings beyond just the laboratory. And this is the promise of Easy CRISPR, to be able to enable high-level, highly accurate molecular detection beyond just the clinical lab. In the same way we think about diagnostics in terms of where we are today and where it should be. Beginning with syndromic detection, a lot of this work at Mammoth was enabled by key partners who helped us accelerate the development of CRISPR diagnostics platform in the context of the pandemic. So we worked with GSK, our government collaborators at NIH and DARPA to really push forward something that was totally new at the time. We are also thinking constantly about how do we continue to advance the capabilities of diagnostics to allow us to detect single nucleotide polymorphisms, to open up faster, cheaper, better genetic testing. How can we enable quantitation to allow us to get very specific levels of detection that open up new and more accessible ways of testing for transplant applications, and of course thinking about how do we lower the barrier for detecting particular diseases in oncology field as well. A lot of the technologies that exist today are serving a very important need, but also there needs to be better and more accessible ways, faster and cheaper diagnostics to really democratize what's possible with nucleic acid detection. So I just want to end with this timeline of some of the early days of the pandemic, just to give you a sense of the different things that were going on at the time. So, as you all know, just before the new year of 2020, China reported a cluster of pneumonia cases. And I remember attending a conference around this time in DC talking about biothreats. And a lot of leaders from FDA, CDC, WHO were talking about the novel coronavirus, and there was a lot of uncertainty about what this meant. I think there was a clear consensus that this was a growing virus with serious concern, and that we needed to make sure that the agencies were prepared to handle this virus with potentially pandemic concern. And interestingly, at the time, I had been working with our scientific advisory board member, Dr. Charles Chu at UCSF, who's an infectious disease specialist, and we had been having these conversations about, being able to repurpose the CRISPR diagnostics platform to address SARS-CoV-2. And during this time, the first confirmed case in the U.S. was identified. And days after that, Charles and I decided to collaborate to develop a CRISPR-based COVID test. And Charles was one of the first physicians in the U.S. who had access to COVID patient samples. As some of you may remember, the cruise ship that had docked in Oakland, and those early COVID cases, Charles had access to the samples. And so within days, we had ordered reagents, tested them on contrived samples at Mammoth, and as soon as some of those first confirmed patient samples arrived in Dr. Charles Chu's lab, we were able to test our CRISPR COVID assay on those and confirm the detection of the virus in those patient samples. And so on the right here is just an example of the visual detection of SARS-CoV-2 using CRISPR on these patient samples, and very quickly we published our work in Nature Biotechnology demonstrating this new method for molecular detection, and we're the first to use CRISPR to detect SARS-CoV-2 in patient samples. And so that was an incredibly intense time for the company and teams working around the clock to address what was a very concerning time and being able to play a part in the fight against the SARS-CoV-2 COVID-19 pandemic. And I just wanted to end with a slide in terms of where I see the opportunity not just for Mammoth but also for CRISPR and the continuum of care. We often think a lot in healthcare about acute care, but the reality is, as people, we are always living on a continuum from health care to sick care, and I think a lot of our healthcare system is focused on acute situations. But I think one of the visions that I have for healthcare broadly around the world is to really allow us to think about not sick care, but really healthcare. And so CRISPR, as you can see, across this continuum from well-being to pre-acute to acute, post-acute, and home care, can play a role in that journey to really improve, what preventative and personalized patient care would look like. So this is not just treating the disease, but early detection and then monitoring, those diseases so that we can spend more time actually being in this wellness and healthy living. With that, I just wanted to again thank all of you for your attention. Tremendous thank you to the entire Mammoth Biosciences team, our partners, advisors, and the opportunities that not only I've been grateful to have, but also what I hope to bring to others as well. So with that, I'm happy to take any questions. Thank you.
Thank you, Dr. Chen, for that wonderful speech. So we are now going to commence a short 15-minute Q&A session. If you guys have any questions that you'd like to ask, please scan that QR code, and then you can ask your questions or vote on existing questions. And we'll just ask the top 4 or 5. All right, so our first question is, what are the scientific boundaries for CRISPR right now, as well as what are your opinions on the moral aspects of this technology?
It's a very good question. There's still a lot of science that is being worked on with CRISPR, and this is involving the safety of CRISPR for gene editing, efficacy in humans, but also just the different ways that you can use CRISPR, right? Beyond just cutting a gene, but also being able to modify single bases, being able to change the expression of genes in different and unique ways. So that's on the scientific side, but also being able to actually deliver CRISPR enzymes, as I mentioned, to specific areas of the body that requires science beyond just focusing on the CRISPR enzymes, but that's thinking about all the things that go around the CRISPR enzyme to actually make this a therapeutic. So there's a lot of very exciting research going on right now in terms of just very creative ways to use CRISPR as sort of the scaffold to bring different types of activities to particular areas of the genome. On the questions, the moral side, I think this is more of the ethics, right? The bioethics of CRISPR. This is I think an evolving conversation. As many of you know, in the last couple of years, there was the event where a scientist had edited twins for a particular CCR5 gene to try to eliminate the potential of those twins developing HIV, and that created a bit of an uproar because there was a lot of conversation about was there really an unmet medical need for doing that and being able to edit these twins that produce outcomes that could potentially be passed on to future generations. So this entire idea of germline editing is hugely controversial because it's still the early days of this technology. We're still trying to understand what's the long-term effect of editing in people and whether or not those edits will be passed on to future generations. And so right now, the scientific community has created a consensus that germline editing should not be done until this technology is better understood, and really just focusing on somatic editing where you can create these genetic edits that wouldn't be passed on to future generations. They're not permanently encoded in germline cells. So that's a big part of it. I think the conversation continues to evolve. There's still the right level of governance that needs to be done And I hope that, the scientific community can hear from all kinds of stakeholders, from, the public opinion to, patients, to caregivers, and everyone who has a voice in what it means to be able to provide this kind of genetic medicine.
So our next question is, how do you identify target sequences for CRISPR in the context of disease diagnostics or therapeutics? Are they mostly in coding or non-coding regions?
Also a very good question. It depends on, I think, the indication. So if you think about it in the context of infectious diseases, oftentimes you're trying to specifically target one virus or bacteria and avoid other similar targets. So if you take just SARS-CoV-2 as an example, there's all kinds of coronaviruses, many the common cold that circulate seasonally. And sometimes there's areas of that virus that look similar. And so if you're trying to think about a target specifically for SARS-CoV-2, you would want to identify parts of the genome that do not have similarities with some of these other viruses. In the case of, let's say, therapeutics, you want to target sickle cell disease. Well, sickle cell is a very well-characterized genetic disease, and there's specific mutations that have been identified that if you correct those mutations, you can reverse the disease. And so in those cases, it's quite targeted, and it really requires understanding of the biology of the disease to actually produce that genetic outcome. So it really, I think, is pretty disease dependent and depending on that use case.
So our next question is CRISPR technology expensive? Roughly how much does it cost? And how will the scientific community work to address the problem of accessibility with technologies like this?
This is a very important point because with any new technology, the scientific community has to think about how do we make these medicines accessible. Right now, there is no CRISPR drug on the market. Right, there are several CRISPR clinical trials that are ongoing, and I think pretty soon, we will, within the next couple of years, we will have the first CRISPR drug that's approved and on the market. So right now, there's currently no precedent for the price of CRISPR drug. However, there is precedent for gene therapy and other types of genetic medicines like, antisense oligos. So I think some of the most famous examples are, Zolgensma and for spinal muscular atrophy. And we know that the price tag for some of these drugs are in millions of dollars, which, as you can imagine, is out of the price range for a lot of patients. However, we have to think about, this is a very complex topic around, how drug pricing is established. So, I'm not an expert in this space, but I think when you are, as we think about how do we actually enable, accessibility of these drugs, a big part of it is, when a first technology is produced and a product is made out of it, it's probably going to have a fairly high price tag. But as we continue to develop new drugs and be able to provide alternatives, there is going to be pricing pressure and competition. And at the end of the day, companies and other institutions are going to, in my opinion, have to bring down the pricing in a way that makes sense for both patients, but also the folks that are putting the capital in to make drugs happen. The process of drug development is incredibly expensive. There's lots of failures. And so that's one of the things that has to be accounted when it comes to actually making a viable drug product. But I believe that over time, pricing is going to be a really important conversation that's going to be hotly debated, especially as we get some of these new CRISPR technologies as therapies to the market.
All right, our next question is, can CRISPR technology be used to help species on the brink of extinction?
Yeah, so one of the interesting stories from Mammoth is that, one of the reasons why we're called Mammoth Biosciences is because we were inspired by George Church, who's a professor at Harvard, and some of his lab's early efforts to resurrect a woolly mammoth with CRISPR. So there was a little bit of a tongue-in-cheek reference when we first were naming the company. And in fact, George Church has now launched a company specifically to use CRISPR to try to focus on this problem of de-extinction. So the short answer is yes, CRISPR can potentially be used to address the extinction. However, I think it's going to be a very challenging problem that's going to take quite a lot of time and research for us to understand, how can we go, for instance, the elephant and actually change the elephant genome to generate, say, the woolly mammoth. So very excited to see this space evolve, and yeah, this is an example of one of the crazy ideas you can use with CRISPR.
How much of your research is based on understanding computational methods versus traditional life sciences or wet lab methods?
I think as technology evolves, and especially for biology and life sciences, it really is an integration of both the computational and the wet lab research. I think we're in a time where the two are very closely intertwined, and while we have people who are experts on the computational side or experts on the molecular biology side, the reality is that those expertise, they have to crosstalk and they have to speak the same language. I think as hopefully aspiring scientists here, you are able to have the exposure to new technologies, both computationally as well as what's going on within the wet lab and new tools. And especially with AI-driven and enabled opportunities, this is going to continue to evolve and very quickly too.
So regarding Mammoth Biosciences, where did you guys get the funding for your company and the research?
Yeah, so we are a VC-backed company, and so the early funding, has been supported through venture capital. We also have, as I mentioned, some collaborations with government agencies and providing non-dilutive funding to help advance some of these projects, and of course, working with partners is another source of funding as well for the company. So that's being able to build a business does require a lot of capital, and we're very fortunate for the support of our investors and partners, both government and private.
So we have a lot of young people in the crowd today, so could you share what prepared you well in middle school and high school to make you become who you are today?
Yeah, I mentioned that I think a big influence on my interest in science, of course, I think came from my dad. So I was very lucky to be growing up in an environment where, science was just a part of dinner conversations, and just the passion that my dad had for science very much influenced me in the early days. And then, of course, having fantastic teachers who didn't focus on the rote memorization of science, but really made me think about what is it about science that allows us to understand more of our world, right? So thinking it more in the context of the scientific discoveries and freedom that really opened up my eyes to what research is. I think it was more than just being in a lab and pipetting. I had mentors who really kind of pushed me and said, okay, help me understand what genes might be involved in this pathway, and how would you answer that question? So even as a pretty early scientist, I was put in situations where, I didn't know how to answer those questions, but I was given the tools to figure it out. And I think that's a really important part of, the scientific process and method, and having the right mentors to put you in situations where you have a safe space to explore, but you also perhaps not, a safe space does not mean a comfortable space. That's one thing I've really have embraced, and I think that's what allows us to really push ourselves to be better.
So coming back to the science aspect a little bit, what are the functions of Cas 12, 13, and 14?
So Cas 12, 13, and 14 are new families of CRISPR proteins. Cas 12 is not just one protein, it really encompasses thousands of proteins, same with Cas 13 and Cas 14. So very broadly speaking, Cas 12 and Cas 14 target DNA. Broadly speaking, Cas 13 targets RNA, but there's a lot of nuance within those enzymes as well.
How do you estimate that CRISPR will affect the next generations of the human race, and what will we do as a community if it causes undesirable effects?
I think the first focus for CRISPR is going to be treating diseases with unmet medical need. Part of that is because of the governance that our regulatory bodies are putting into place, and of course, the scientific community in driving, where the effort and the funding should go. And there's, thousands of genetic diseases that have no permanent cure. And I think we could spend, probably the rest of my lifetime even starting to address that. I think, in the future, it's a big question, and I think there's still a lot, that needs to be understood. I think there's, oftentimes you'll hear in the media about using CRISPR to, edit IQ or appearance, physical traits, things like that. And the reality is that we're quite far from even understanding, how you would even do that. And my strong belief is that there are so many areas of unmet medical need that we'll be focusing the technology on it, especially in the near future.
So I think this is going to be our last question for this speaker event. In which situations is Mammoth Biosciences COVID recognition methodology being used right now?
In which situation? So with COVID-19, we had worked with the NIH to develop a laboratory high-throughput CRISPR COVID test. And so this was really to increase the throughput of testing for laboratories that were completely underwater by the number of patient samples that they were being received. And so, through the NIH and in conversations with the FDA, we had received EUA for our high throughput COVID test. And so this particular application was for use in laboratories.
Maybe we have just a couple more minutes, so one more question. How did you choose the area of research that you went to?
I was always just following what I thought was exciting and interesting. As I mentioned, I think molecular biology has always been very exciting because of this programmability aspect. And, not surprisingly, when I was looking at different graduate programs and labs, CRISPR is a programmable tool. And the idea that you could program biology or engineer biology to, do some very interesting things is a theme that I think has carried throughout, my early scientific career to where I am today. And I think there's just so much you can do in terms of engineering biology.
Thank you so much, Janice, for speaking with us here today. With that concludes our opening keynote address. Sorry. And from here, you guys are free to check out the student speakers in the Nichols Auditorium. We'll also have exhibitors like our student robotics and from 12 different companies. We'll have STEM buddies in there, which are basically science activities for our younger students. And we'll have some poster sessions in the auxiliary gym and another keynote address here at 10:30. Thank you guys so much for coming today.